The urease test is used to test to see if bacteria have an enzyme called urease, which is an enzyme that is able to hydrolyze urea or break down urea into ammonia and carbon dioxide. This test specifically is used to differentiate between bacteria that are urinary pathogens. So there's a lot of different bacteria that can cause infections like urinary tract infections or UTIs, because the urinary tract is supposed to be sterile at all times, so anything that potentially gets in the urinary tract could lead to an infection. So a lot of different bacteria can cause UTIs. So a lot of times we'll have to differentiate between what the potential causative agent is for treatment. So a urease test can be used to help us kind of figure out what type of bacteria we're looking at, because bacteria like Proteus, which cause UTIs, they have the urease enzyme, so they would cause a positive reaction to happen during this test. However, bacteria like E. coli, which are also can cause UTIs due to kind of fecal contamination, they don't have the urease enzyme, so they would produce a negative result here. So really, this test is used for kind of differentiation between urinary tract pathogens. So in this media, we have urea. So urea is the primary component of the media, but also within the media, there is a pH indicator called phenol red, which at a neutral pH is this orange color. So these test tubes, before they've been inoculated, they're just naturally this orangish color. So when you inoculate them with bacteria, if the bacteria has the urease enzyme, it will break down urea, which causes ammonia to be released, which causes the pH to increase. And in these test tubes, because of the pH indicator, phenol red, when the pH increases, the color shifts from orange to this bright pink that we see over here. So in a urease test, if the test tube turns bright pink, that's a positive reaction, and it means the bacteria have the urease enzyme and are able to hydrolyze urea. However, if you put a bacterium in here like E. coli that lacks the urease enzyme, urea is never broken down, which means ammonia is not released, which means the pH does not change. And since the pH does not change, the color does not change, and it stays orange. There are some test tubes that will turn yellow, but either way, orange or yellow, still both a negative result, meaning the bacteria do not have the urease enzyme and are not able to break down urea. So in order to do a urease test, the urease test is a liquid broth. So you would take an inoculating loop, you would sterilize it, you would let it cool down, you would collect a sample of bacteria, and then you would just mix the sample of bacteria into the broth solution, and then you would obviously sterilize your loop afterwards, but then you would incubate for 24 hours. We typically need to read this test within 24 hours if we're looking for rapid urease breakdown, or rapid urease production, which leads to urea breakdown. But within that 24 hours, if the test turns pink, that's a positive reaction. If the test stays if this test stays orange, that's a negative reaction. The next test is the citrate test. The citrate test is used to see if bacteria are able to use citrate as their sole source of carbon. And carbon is an essential nutrient. All organisms need it to survive. And the only carbon source in these test tubes is citrate. So if the bacteria is able to use citrate as a source of carbon, it will do so in order to survive. However, if it's not able to, then the bacteria that are inoculated in these test tubes will ultimately die. So what we're really looking to see is do our bacteria have enzymes that allow them to utilize citrate as a sole source of carbon? And those two enzymes that we're testing for are citrase and citrate permease. So if bacteria have citrate permease, that means that they're able to take citrate into the cell through their cell membrane. And then if they have citrase, that means that they're able to break down the citrate and use it as a source of carbon. And then obviously bacteria that lack these enzymes would not be able to use citrate as a source of carbon. So essentially they would not be able to survive in this type of media. So the citrate tests are what we call agar slants. So it is a solid media in a test tube that runs on a slant. And in this media, there is obviously citrate. That is the sole source of carbon. So we want to see if the bacteria have the enzymes to utilize it or not. And then there's also a pH indicator called bromethyl blue. So how it works is if you put a bacterium in here that has the citrase and citrate permease enzymes, it will be able to utilize citrate as a source of carbon. And when it does so, ammonia gets released, the pH goes up, and the pH indicator causes the media to turn this kind of bright blue. So blue is a positive result for the citrate test. However, if we put a bacterial sample in here that lacks the citrase and citrate permease enzymes, meaning it cannot use citrate as a source of carbon, then ammonia never gets released, the pH does not change, and the color does not change. And in that instance, it would be a negative result. So in the citrate test, blue is positive, green is negative. So to do a citrate test, you typically do want to use an inoculating needle so that you're not adding too much of a bacterial sample, which can sometimes confuse the results. So what you would do is you would take your inoculating needle and sterilize it, let it cool down, collect a sample of bacteria, and then you would really just kind of zigzag that sample of bacteria up the slant. You don't stab this media like you do in the SIM test. You really just kind of zigzag it up the surface of the slant. And then you incubate for 24 to 48 hours and then come back and look at your results. If it's blue, that's a definite positive result, meaning your bacteria has the citrase and citrate permease enzymes and is able to use citrate as its sole source of carbon. If it's green and there's no growth happening, then that is a definite negative result. However, sometimes there will be no color change, meaning it's still green. However, there is growth. In cases where it didn't turn colors, it's still green, but we do see growth, that is still interpreted as a positive reaction. Sometimes due to errors with incubation, the color change won't happen, or if you don't add enough bacteria, the color change won't happen. However, in those cases, if we can see active growth like what we see here, we still want to interpret it as a positive reaction. So really there's kind of two positive reactions, either the definite blue shift or it remained green but had growth. Both of those positive. But definitely if it's green and there's no growth, then that is a conclusive negative reaction.